All right, let's talk about something that's, well, it's completely rewriting the book on biology and medicine. We're jumping into what's called next generation sequencing, or NGS. And it's taking us from just, reading the letters of life's code to actually understanding the language itself. Pretty cool, right? Let's dive in. Seriously, just stop and think about that for a second. What if your doctor could just pull up your entire genetic blueprint to predict, diagnose, and treat diseases with absolute pinpoint accuracy? And look, this isn't some far-off sci-fi dream. This is the reality that NGS is building, like right now. Okay, so what is this stuff, really? Well, think about it like this. The old way of doing things, Sanger sequencing, that was like painstakingly reading a single book, one letter at a time. I mean, it was incredible for its day, but man, was it slow. NGS? That's like having a million tiny robots scanning and digitizing the entire Library of Congress, all at the same time. The jump in speed and scale is, well, it's just mind-boggling. It's a total revolution. So let's get into our first section, the genomic revolution. And we're calling it a revolution for a reason. This isn't just a small step up, it's a complete paradigm shift for biology. The impact? It's huge. Just think about it. Scientists can now hunt down the exact genetic typos causing rare diseases in record time. Or they can map out a tumor's entire genetic weak spots to create custom-tailored cancer treatments. And we all saw this in action, right? Tracking a virus like COVID-19, watching it mutate in real time. That's NGS at work. It's an absolute game changer. Okay, Section 2, meet the technologies. So how does all this magic actually happen? Let's get into the nuts and bolts and look at the machines that are making it all possible. So here's the thing. NGS isn't just one magic box. It's really a family of technologies, and they kind of fall into two main camps. You've got what's called second-generation or short-read sequencing. This is the established workhorse, super accurate. And then you have the newer kid on the block, third-generation or long-read sequencing. This one's all about portability and reading these huge, unbroken chunks of DNA. Which one you use, it all comes down to what question you're trying to answer. First up, the undisputed king of the hill, Illumina. This is the short-read tech we were just talking about, and it completely dominates the market. Why? Because it spits out billions of DNA snippets that are incredibly accurate. We're talking over 99% accuracy. This makes it the go-to for big research projects and for most of the clinical stuff you hear about, like sequencing a patient's entire genome. It's the gold standard for a reason. But then you've got the trailblazers, right? Companies like PacBio and Oxford Nanopore. These are the long-read guys, and they do something pretty wild. They can actually read a single, super long strand of DNA without chopping it up first. And Oxford Nanopore has even made sequencers so small they can fit in your hand. You can literally take it out into the field to identify a new virus on the spot. It's genomics on the go, happening in real time. All right, next up, Section 3, decoding the data. Because, getting all this sequence information is one thing, but it's just a giant pile of letters. The real challenge is turning that into something meaningful. And that happens through something called a bioinformatics pipeline. It's basically a three-step process. First, the machine spits out all the raw genetic data. Then some seriously powerful software gets to work. Think of it like a massive digital puzzle, where it takes all those billions of little pieces and lines them up against a reference map of the human genome. And finally, once it's all lined up, algorithms flag any differences, the variations, and that's when scientists step in to figure out, okay, what does this specific change actually mean? And the amount of data we're talking about here is just staggering. It's not a river of data, it's a full-on tsunami. A single run on one of these machines can produce terabytes of information. That's an enormous problem to solve from a computing standpoint. It was a huge bottleneck for a long time. So, how in the world do you handle a data tsunami? Well, you need even smarter tech. We're talking about leaning on cloud computing for storage that can scale up as needed. We're using the same powerful GPUs that run your video games to crunch the numbers way faster. And here's where it gets really cool. We're bringing in machine learning and AI. These algorithms can spot patterns no human ever could, improve the accuracy of the data, and even weave together different types of biological data to get a much richer picture. Okay, on to Section 4, big challenges, bigger questions. Because look, with all this power comes, well, great challenges. And it's not just about the tech. We're also staring down some really big ethical questions. Let's start with the technical hurdles.
First, data storage. We talked about the data tsunami. Where do you put it all? It costs a fortune. Then there's the raw computing power needed to analyze it all. It's intense. But maybe the biggest bottleneck? It's people. There just aren't enough trained bioinformaticians who know how to manage and interpret all this complex data.
But, the tech problems are almost the easy part. The really tough stuff is ethical. I mean, think about it. Who actually owns your genetic code once it's been sequenced? And what happens if, while looking for one thing, a doctor finds a gene that suggests you're at high risk for a completely different disease? What's the right thing to do then? This is a real ethical maze. It starts with patient consent. People have to really understand what they're agreeing to. Then there's the issue of those incidental findings. We need clear rules for how to handle them. And of course, data privacy is everything. This is maybe the most sensitive information about you that exists. It gets even trickier when you try to share data for research across countries, bumping up against privacy laws like GDPR. None of this has an easy answer.
Okay, final section, number 5, the future of medicine. Because if we can navigate those challenges, the payoff it's just incredible. We're literally looking at the future of how we practice medicine. Check this out. One of the bleeding edges right now is a field called spatial transcriptomics. This is absolutely wild. It's not enough to know which genes are turned on in a piece of tissue. This tech lets us see exactly where in that tissue they're active. It's the difference between having a list of ingredients and having the full 3D architectural blueprint of a disease.
And hey, remember those little handheld sequencers we talked about? That's leading directly to what's essentially a clinic in your pocket. Just picture it, a doctor in a remote village or at a patient's bedside getting complex diagnostic results in minutes, right from a device in their hand. That's where we're headed. So when you put it all together, what does this next wave of healthcare look like? It looks like truly personalized medicine with drugs designed for your genome. It means spotting outbreaks almost instantly. It means discovering new drugs way, way faster. And it means watching a cancer treatment work in real time and adjusting on the fly. This isn't just a promise anymore. It's happening. So I'll leave you with this. Genomics is handing us the keys to the kingdom, the ultimate blueprint for life itself. The tech is moving at a dizzying pace. We can read the blueprint. The bigger question is, are we really ready for what we're going to find when we do?